Hi, and welcome to the Arts and Sciences Week. This week, we're showcasing our faculty and students within arts and science, the broad range of work and activity that is being done. A couple days ago, we had a showcase of undergraduate students talking about their work. Later today, we have at 3 o'clock, we have a showcase of graduate students in arts and science talking about their work. But right now, we're very fortunate to have a number of faculty in arts and science who are going to be talking about their work. And so we have a number of folks here that we're going to work our way through, and each one will have an opportunity to talk for about 5 minutes. Let me introduce who we have, or who we will have here. We have Lindsey Davis from Humanities and Arts, Laura Ficara from Robotics Engineering, Stacy Shaw from Learning Science and Technology, Alex Smith from Social Science and Policy Studies, Sam Walcott from Mathematical Sciences, Kunta Wu from Physics, Ali Yusefi from Computer Science and Neuroscience, and Liz Ryder from Biology and Biotechnology. So all of our faculty are with us. And so what we're going to do here is when I turn it over to the first speaker, we're going to go through each speaker will give their presentation. Okay, you are welcome and in fact encouraged to enter questions while the speakers are talking. But we're not going to stop. We're going to make sure we go through all the lightning talks of each faculty member. But once we get through all the talks, then we'll open it up for questions across all faculty. And at that point, we'll go back to the Q&A and we'll go through those. So make sure that you're entering your questions as we go through, or you're certainly welcome to do that. Okay, so with that, I'm happy to introduce Lindsey Davis as our first speaker.
Okay. First, I'd like to thank Dean Jean King and Dr. Kate Moncrief for asking me to be a part of this esteemed panel of scholars. I'd also like to express my gratitude to my fellow panelists and to Craig for facilitating this conversation. And I'd also like to thank Rebecca Ouellette for all of her behind the scenes support. So this talk is titled White Feminists, Black Squares, and Performative Activism. And it's part of a larger project, which is still in its infancy, that analyzes the role that social media influencers play in the context of feminism, whiteness, and capitalism. So the social media influencer industry is, at a conservative estimate, a $15 billion business that really tends to kind of highlight and center the work of white cisgender women. Many began as fashion, lifestyle, and/or mommy bloggers who were looking for creative outlets outside of their 9-to-5 jobs. With the advent of Instagram in 2010, many of these bloggers, such as those you see here from left to right, Carly Reardon of Carly the Prepster, Grace Atwood of The Stripe, Emily Schumann of Cupcakes and Cashmere, and Naomi Davis, no relation, of Love Taza, they expanded beyond the confines of their blogs and began to monetize their lives as mothers, women, and in time as kind of micro celebrities. Today, they curate sponsored posts for J. Crew, they host events for American Express, create limited edition collections with multinational companies like Target and Amazon, and they earn six-figure salaries. Social media influencers such as these are part of an industry undergoing exponential growth, something that has expanded from $8 billion in 2019 to $15 billion, as I mentioned, projected in 2022. In the wake of the murder of George Floyd and the subsequent Black Lives Matter protests, white influencers such as these found themselves at a bit of an impasse. Their idyllic social media content suddenly seemed at odds with the news headlines, and it became increasingly untenable for them to post things like sponsored content for Starbucks or the perfect cheese plate when people around the country took to the streets to protest the murders of unarmed Black and Brown people. Political discourse and activism should and is nuanced, and the influencer aesthetic just doesn't allow for that. So while many of these influencers hinted at progressive and feminist leanings, their Instagram feeds featured posts at subtle odds with those priorities, as well as increasingly critical comments from their followers, pointing out that influencers appeared at best to be out of touch and at worst complicit in holding up structures of white supremacy. On June 2nd, two music industry figures organized a social media-based collective action, which was known as Blackout Tuesday, to protest the murders of George Floyd, Breonna Taylor, and Ahmaud Arbery. Influencers, among many others, joined the effort, posting a single Black square in their feeds with the hashtag Black Lives Matter. Many included accompanying captions or reflections, such as Mormon mommy blogger Naomi Davis of Love Taza, who wrote, "For a long time, while conversations surrounding racism came up, I'd just say or think, well, I'm not that, and leave it there. But that's where I've been wrong, where my privilege comes into play. I have to be more than, quote, not racist." Though the intent was to mute white influencers, the utilization of the Black Lives Matter hashtag actually had the opposite impact. And instead, it failed to decenter white voices and clogged the feeds with Black squares after Black square. After Blackout Tuesday, many influencers continued to utilize a kind of bubblegum hued version of feminism or progressivism to express their solidarity, posting stylized quotes from Ruth Bader Ginsburg or Desmond Tutu, highlighting their anti-racist reading lists, and spotlighting Black-owned businesses. Yet most struggled to meaningfully navigate the terrain, to successfully navigate the terrain of anti-racism. For many, this content was relegated to the ephemeral Instagram story function, which disappears after 24 hours. As savvy creators, it is possible that some of these influencers opted for this type of temporary approach to both limit negative public comments and avoid the permanence of what might be deemed potentially controversial content. Yet, why is this important? Why should we care about the political actions or inaction of social media influencers? For one, many of them have massive audiences ranging from 100,000 to over 1 million followers. Two, studies show that a majority of consumers consult influencers for purchasing advice, voting recommendations, product recommendations, and guidance on topics ranging from the best cleaning products to medical procedures. When they partner with companies facing allegations of hostile work environments from women, people of color, and other underrepresented populations, their personal brands uphold destructive practices of sexism and white supremacy. And three, history is littered with examples of the negative impacts of white feminism, particularly when white feminists fail to reckon meaningfully with structural racism and white supremacy. Social media influencers continue to traffic in the rhetorics and aesthetics of this particular mode of white feminism to define their brands, court girl power oriented corporate partnerships, and appeal to like-minded followers, and their reach is vast. However, history tells us that a feminist approach that papers over intersectional concerns about race can be destructive. And instead, it is vital to center and recenter voices of color, particularly in the influencer space, as a way to reshape these conversations and pave a more productive anti-racist path forward for the contemporary feminist movement. Thank you so much for your attention. I look forward to any comments or questions at the conclusion of these talks. And with that, I will turn things over to Loris Ficara.
Okay, good afternoon. So I'm a surgical roboticist, and today I would like to talk to you about one of the robots I'm currently building in my lab at LPI, whose goal is to treat the vocal folds using laser light. It is only fitting that I get to do this today, since April 16th is World Voice Day. So let me tell you first why we need robots to repair the vocal folds. People who make a professional use of their voice have a very high risk of developing vocal folds damage. These individuals include actors, singers, news anchors, scientists, especially those who speak in public very often, like Dr. Fauci of the NIH. College professors are also at risk, and educators in general. And the list goes on. Possibly it is longer than you may reasonably expect. Research tells us that about 3% of the general population at any moment in time suffers of some form of benign laryngeal lesion, as doctors call them. Now, the good news is that doctors know how to treat these lesions, and very frequently the treatment relies on a surgical procedure. The not so good news is that vocal fold surgery is a very delicate procedure. It needs to be carried out under general anesthesia. It is not without complications, and it can be really expensive. So for the past few years now, doctors have been trying to explore ways to keep vocal folds treatment out of the operating room. And this is what they came up with. They came up with this idea of performing vocal folds surgery in the office as an outpatient procedure. And this is how it is performed. Doctors utilize a flexible, tiny endoscope that is passed into the throat. And then with the help of a very tiny optical fiber, they apply laser pulses on the vocal folds, trying to thermally destroy this tissue that has to go away. This procedure, as you can tell from the figures, it can be carried out without general anesthesia. It typically only takes a few minutes. It is much, much less expensive than surgical treatment in the operating room. In fact, it costs only 1/4 of traditional surgical treatment. And it is relatively straightforward to perform. So I remember when I started learning about these procedures, I was asking myself, well, if there are so many benefits, why is it that in 2020 and in 2021, people still need to be seen in the operating room for vocal folds procedures? If you read the medical journals, doctors say that there are some limitations in the instruments that they have available right now. It's very difficult, apparently, for them to aim the optical fiber on target. And there are certain parts of the voice box anatomy where they really have a hard time reaching. I am not a doctor, so I wasn't able to relate exactly to what they were saying, but I really wanted to understand more. And so what I did was to do what any other engineer would have done. I coded up a simulation together with my students, and using techniques that we normally use to simulate robots, we simulated an endoscope with an optical fiber within a realistic model of the human larynx. I say realistic because what you see on screen right now, this blue shape, is actually the true anatomy of a real person. And assisted by a clinical collaborator, we reproduced in the simulation the same range of motion that a doctor would perform during a surgical procedure. And then we let this simulation run for several hours with the intent of building a map of all the places where the doctors are able to go. And what we found is that if you look at this figure here with all these red dots representing locations that are accessible by the doctors, there is a wide open gap right here in the middle. We showed this to the doctor and he said to us, yes, this is exactly the location where I'm having a hard time reaching, and this figure visualizes that. We submitted these results into a conference paper earlier last year together with my students, and they won the Best Student Paper Award. I think one of the merits of this paper is that it is a very good example of how we can use engineering tools and computer science tools to bridge the gap between medical knowledge and make it explicit for engineers and computer scientists. So now we can very much understand where doctors have a hard time reaching. And to solve these challenges, we came up with this. We are building a steerable robotic laser probe that is going to enable doctors to point the laser fiber onto those locations that are currently beyond reach. And for this project, I partnered up with Tom Carroll, who is a physician and the director of the otolaryngology clinics at Brigham and Women's Hospital in Boston, and my colleague, Sean Liu at WPI, who is an expert in laser optics. One of the challenges of building this instrument is that it needs to be really tiny. Here is one example of the first prototype that we built. It is only 1 millimeter in diameter, so we are really approaching almost needle size here. But fortunately, we have the expertise to do this at WPI. And so we're looking forward now to doing some preclinical tests. Thank you for watching. I'll be happy to answer any questions you may have. And now I will pass it on to my colleague, Stacy Shaw.
Thanks. All right. Hello, everyone. My name is Stacy. I'm a new faculty at WPI in the Learning Science and Psychological and Cognitive Science area. And today I'm here to talk to you about understanding the aha moment of insight, specifically within mathematics. So aha moments are these eureka moments where an idea comes to us that's sudden and unexpected. And they're particularly interesting in mathematics because they tend to signal that learning is taking place. So math students, for example, can have aha moments when they finally figure out how to solve a problem or make a connection to a new concept or come up with a creative idea. So as a learning scientist, I'm really interested in understanding how these aha moments occur. In the past, what I've done is I've brought university students into the lab to study this, and I give them this math insight puzzle. So let's say you're a participant in my study, I bring you into the lab and I show you these 4 digits and 2 symbols. And I ask you, can you come up with a way to rearrange these digits and symbols in a way to make a balanced equation? With the caveat that you have to use each of the digits and symbols once and only once, and you can't add any symbols or digits or take any away or duplicate. And what we tend to see happen is students really struggle, right? They try and make larger numbers, they try and combine them in different ways, and very quickly they realize that on the surface level, this is very frustrating, right? It doesn't make sense, you cannot solve the problem this way, and they agonize over trying to find a solution to this problem. But you're not gonna do that because I'm about to give you the solution. So spoiler alert, to solve this problem, you really have to think outside the box, right? You have to think past addition and think of exponents to see that too as an exponent. So 3 squared equals 4 plus 5. And what I found through my research is that most students have a really hard time solving this in the laboratory. In fact, only about 20% of them are able to solve in the lab, and 80% of them don't. So if you're a participant who can't solve this problem in the lab, you're lucky enough to get enrolled in my second phase of the study, and it looks something like this. So you don't solve the problem in the lab, what happens? I kick you out of the lab, right? I say, go live your life, but here's an email with a link to a survey. Now I want you to go ahead, live your life, business as usual, but if you happen to have an aha moment, go ahead, open up that survey and tell me about that aha moment. And what I found is about 50% of students who normally can't solve in the lab go on to have these aha moments in their everyday lives. In fact, we saw examples of all those classic aha moments happening in the shower, walking the dog, that sort of thing. But what's really interesting is I also found evidence for this very fascinating theory of, sort of what creates these aha moments. And that theory suggests that what's actually happening is once you fail to really solve a problem and then you go somewhere else or you go for a walk, you've become sensitive to information, incidental cues in your environment that can help you solve. So it's sort of like once you've really struggled, you have a guide in your head pointing out important landmarks that might help you solve. So let me give you an example. One student who solved said, I was in class and the main topics were form and power. And that's when it hit me that taking the power of a number doesn't require another symbol, and I smacked myself in the face, right? So this is an example of a student who happened to run across the word power and made that connection. But not all of these incidental cues in the environment are that concrete. For example, another student said that she was on the phone with her boyfriend and he was rambling on about a Minecraft stream or something, and she started zoning out looking at the corner of her desk. And it made her think of right triangles, which made her think of Pythagorean's theorem, which made her think of a squared plus b squared equals c squared, and she made the connection to exponents. Now, what's really interesting about these two examples are that they are sort of representative of students who were conscious. They were aware that they had these hints in the environment that helped them solve. But this theory is really suggesting that most of the time this happens at an unconscious level, outside our conscious awareness. That's why aha moments feel like they come out of nowhere, because we're just not aware of these hints in our environment. So after the study, I grabbed 10 unsuspecting undergrad students and I plopped them in the lab and I gave them this math problem, and they all failed. And then what I did, instead of kicking them out of the lab, I had them interact with this poster. And you could see there's a very heavy-handed Q right there in the middle, E equals MC squared. And I had them interact with them for 2 minutes. Afterwards, 7 out of 10 of them were able to solve this problem. But when I asked those 7 students, was there anything in your environment that helped you solve? None of them could come up with anything. So my future directions, I'm really interested in understanding how to engineer these aha moments, right? How to get students to struggle through educational materials like online textbooks or worksheets and deliver that incidental cue that might trigger that aha moment. And this isn't to say that I'm really trying to get students to learn more through aha moments, but aha moments, some research suggests, constitute the only positive experience some students have in math classrooms. So it's the payoff after working really hard, feeling that like they don't get it, like they're not going anywhere, to have that euphoric moment of understanding. So in my future work, I'm really interested in triggering these moments and then studying how they affect subsequent persistence, motivation, things like that. So in conclusion, more research is always needed to be done, but thank you so much for listening. And next up is Alex Smith.
So my name's Alex Smith, and today I'm going to talk to you about some ideas on collaborative policy design. And the thoughts that I'm going to share today, they grew out of some casual discussions that I was having with colleagues a couple years ago. And really our primary goal was just to find some ways of combining our diverse areas of research interest and expertise. And here's what we came up with, which we think are some pretty neat ideas for how to approach policy design. So if you think about international development following World War II, institutions like the World Bank and the United Nations, they tried to promote policy development around the world, and they had a number of reasons for doing this. They had a genuine interest in the well-being of others, but also there was this view that that improving the standard of living in developing countries would be good for stability, political stability around the world. So there were some good reasons for these efforts at international development. Unfortunately, many of these efforts failed because of a disregard for local context. One common example among many is the Kariba Dam project on the Zambezi River between Zambia and Zimbabwe. By some counts, the project displaced as many as 60,000 villagers who lost their homes as a result of the dam being built. And what's worse is that it wasn't really clear that these people wanted the hydroelectric power anyway. So this is an example of where the policy solution maybe wasn't appropriate to the local situation. Now, in response to lessons such as these, policy practitioners now recognize the benefits of local participation. And, as an example of this, Engineers Without Borders is great at working with people locally to construct things like water wells. And by working with local people, you create sustainable solutions because you've engaged the local people in your efforts. We wonder if this is good enough. And the reason we're not sure that this is good enough is because even with participation, you're still taking a solution from the outside and trying to impose it locally. And it's not always obvious that the outside solution is the best way to go. So what we advocate in a book chapter that we've recently drafted is something that we call the collaboration principle. And what that is full-fledged partnership between outsiders and local stakeholders. So first what you do is you work together to determine the goals of policy. And once you've figured that out, then you can start determining the appropriate policy interventions, which of course is a joint effort as well. And then when you've identified possible policy interventions, we recommend using randomized controlled trials to measure the effectiveness of policy. Now, the aim with this kind of approach is to create sustainable solutions that are possible to sustain long-term using local resources And then also they're culturally appropriate because the solutions have been designed as a part of a joint effort between outsiders and local people. So that's our philosophy regarding how to do policy design. And I want to quickly share with you a couple of ways that we're getting started on this. And the first is Assistments in Africa. Assistments is an online learning platform developed by Neil and Christina Heffernan at WPI. And it's a platform where the main virtue is that you have flexibility to customize content, the questions and problems that students solve, and better yet, you can customize the feedback that students receive. So you can make it appropriate to the local situation. And in the fall, we did a trial in Ghana with the help of Kennedy Demoa, who's a grad student at WPI. And it was a small trial with a small number of teachers in Ghana, but the anecdotal evidence that they reported to us is that the customizability and ability to make the platform fit the local situation was very helpful for them to produce a tool that was good for their students' learning. So the next step there is to do a full-fledged randomized controlled trial to measure effectiveness. And of course, we also want to expand. So we're talking to WPI partners in a number of other African nations about perhaps joining a trial of assessments to test out how well it can work when the full customizability is taken advantage of. And one final example that I'll just quickly touch on that we're just starting to talk about with a couple of other colleagues in SSPS is working with students of color at WPI to co-design policies with the aim of possibly making the campus experiences even better than they already are. So we're taking this philosophy about policy in a few different directions, and hopefully in the future we'll have plenty to share about that, but I'll leave it at that for today. Thank you very much, and I'll be happy to answer questions later. So I'm now going to turn it over to Sam Walcott.
All right, thank you very much. I'm interested in how interactions between molecules affect biological function. I've worked on a variety of biological systems, some of which you see here, but today I'll talk about intracellular transport. Life depends on cells transporting material, like proteins, through their interior. These interiors are complex 3D networks of protein filaments, as you see in the picture in the upper left. Cells move material by packaging it in membrane-bound vesicles, and then molecular motors walk along the protein filaments, carrying the cargo to its destination. On the right, you can see a movie of one of these motors. By studying individual motors, we understand how the chemistry of each foot depends on force and how that allows the motor to coordinate stepping. But how can we relate this knowledge about single motors to what happens in a cell where multiple motors work together to transport their cargo through 3D networks of protein filaments? As a first step in addressing this question, we manufactured 350 nanometer membrane-bound vesicles called liposomes, onto which we attached 10 myosin molecular motors, as you see in the top left. We made suspended intersections of actin filaments, as you see in the top middle image, and then watched as the molecular motors moved the liposome through the intersection, either going straight, as you see in the images at the top, or turning, as you see in the movie below that, or detaching. But we can't see the motors, so it's hard to understand why we observe these outcomes. We therefore made a computer model of the system, including the mechanics of the liposome and actin filament, as you see in the lower left, the mechanics of the motors, as you see in the middle, and the motors' known force-dependent chemistry, which you see in the lower right. This model, if correct, would let us see the individual motors. But how do we determine whether the model is correct? Well, one thing we can do is test its predictions. In the model, although 10 motors move freely over the surface of the liposome, we almost never see more than 3 bound simultaneously to the actin filament. This occurs because motor binding restricts the motion of the cargo, as you see in these movies, making it harder for the motors to reach the actin filament. We tested this prediction using a laser trap to hold onto the liposome as the motors pull against it, as you see in this schematic at the lower left. The motors move the liposome forward and the laser applies a force proportional to the distance, like a spring, as you see here in the bottom middle. The motors move until they stall, as you see here, and that is they can't overcome the force of the laser. Eventually the motors detach, the liposome goes back to the middle of the trap, and the process repeats. Stall force is proportional to the number of bound motors, so by measuring the stall of a single motor, which is about 2 piconewtons, we identified the peaks of 2, 4, and 6 piconewtons in our measurements as 1, 2, and 3 motors, consistent with the prediction of a 3-motor limit. All right. Although we know a lot about the model, there are 2 unknown quantities, one having to do with, sorry about this. We have a little bit of a lag here. Okay. Okay, so although we know a lot about the model, there are 2 unknown quantities, one having to do with how quickly the motors attach to actin, the others having to do with how bendy they are. We picked these values to make the model agree with our observations, as you see in the upper left. And we can then watch what the motors do as the liposome approaches and passes through an intersection, as you see in the movies at the bottom. And note that I'm only showing the motors that are attached to actin. All right, though this is a step in the right direction, the previous experimental system is simpler than reality, where motors move their cargo through a random, complex 3D mesh. So as a next step, we made 3D random meshes, as you see in the upper middle picture. And then we watched as the 10 myosin molecular motors transport the 350 nanometer liposomes through them, as you see in the bottom left image. We then made a computer representation of the mesh. We ran the model on the same 3D networks as our measurements and compared the model to the measurements in various ways, found good agreement. This is my favorite comparison we did. In our experiments, we occasionally observed liposomes pause. That is, they stay more or less stationary for longer than a second. You can see this as the blue dots in each panel. When we ran our simulations on the same geometries, we observed pauses in the same location, as you see in the hollow circles. We looked at this in more detail. I'd like you to focus on the upper right panel. Okay, so what I'm going to show you a movie. And in this movie, there are a bunch of small blue spheres, which indicate points where we saw pause in our experiment. The movie will show a simulation. And at some point, motors will be simultaneously bound to 2 actin filaments, starting a tug of war. When this begins, the center of the liposome will be marked with a red sphere. Here, the tug of war starts. And as you can see, the blue and red spheres are remarkably close. So we conclude that the tug of wars underlie the observed pauses. Having a model that successfully predicts our experiments allows us to extrapolate beyond where it's possible to measure. For example, we can investigate how network density affects the transport of thousands of liposomes, as you see in these movies. On sparse meshes, as you see at the left, liposomes move rapidly in a straight line on 1 or a few filaments. The filaments turn yellow once a motor binds. On dense meshes at the right, the liposomes move slowly and spread out along many filaments. Both behaviors have been observed in cells, and the model suggests that the cell can modify filament density to regulate this. I hope I've convinced you that mathematical modeling is a useful tool to connect molecular scale measurements to cellular function. Hold on a sec, I have one more slide. Here are the people who did the experiments. It was funded by the NIH, and I'd like to turn it now over to Kun-Ta Wu.
Okay, so I was muted. Okay, so thank you, Sam. So hello, everyone. Thank you for coming to my talk. And today, what I'm going to show you is our recent research finding, showing that the active fluid flow within the droplet is coupled to the oil flow near the water-oil interface. And this water-oil coupling will determine the fate of intra-droplet active fluid flow. So what is active fluid? Well, our active fluid system contains three main components. The first component is microtubule, what I show here, which is a protein-based fiber. And the second component is depleting polymer, which will induce a depletion force to bundle microtubules. And this bundle microtubule will be bridged by our third component, kinesin motor dimers. And this motor dimer will slide pairs of anti-parallel microtubules apart, causing interfilament sliding. And the sliding dynamics will accumulate to a microscopic scale, resulting in a percolating gel network whose structure constantly self-rearrange. So this self-rearranging gel network is called active gel, which is our active fluid system. So we confine this active gel in the water in oil droplet and find out that the gel can spontaneously form intra-droplet circulation. So to better visualize and characterize this circulatory flow, we dope the gel with tracers. I find out that the tracer will flow with the microtubules and swirl within the droplet. So this is our experimental setup. So what I want to do now is we investigate what parameters will control the formation and deformation of this circulation. So the first thing we find out that is the circulation is not supported in all droplets. In some droplets, the circulation developed. In another droplet, the circulation is suppressed. So that just shows that the circulation depends on droplet geometry, which makes sense. But what doesn't make sense is that we find out the circulation depends on a parameter that's irrelevant to the droplet geometry, which means that in this, in these two examples. So we have pair of droplets that have a similar shape, but immersed in different oil baths. And we find out that in one oil bath, the circulation developed. In another oil bath, the circulation is suppressed. Why would this happen? Does it make sense to us? So to get a deeper insight, we developed a continuous active fluid simulation based on existing self-elongating rod model. And so surprisingly, our simulation reproduced the experimental outcome. We got circulation in one oil bath, and the separation is suppressed in another. And so the simulation shows that the reason we have this oil bath dependence is because of the flow coupling between active fluid and oil, which means that active fluid can induce oil flow, induce the flow in oil, and the oil flow can also in return influence active fluid flow. So with this finding, this finding suggests that we can just control the formation and deformation of intra-droplet circulation by just disturbing oil flow near the water-oil interface without touching the droplet. Right, so this is the idea. And so to test this idea, we developed a mini fluid device that has a movable wall, which is this pink piece. So using this mini fluid device, we approach the wall toward the droplet and find out that circulation disappeared, so we have we measure the circulation order parameter and COP decrease. And on the other hand, when we move the wall away from the droplet, and the droplet can notice it and recover the circulation. So this result shows that A, our hypothesis is correct. This flow coupling between active fluid and oil is true. And B, we can control the circulation and intra-droplet circulation in real time. We just move the wall back and forth. And C, this droplet is like a child, because we know when the child is the child will behave well when the mommy and daddy is at home, but they will become mischievous and creative when they're away. So we say this droplet has a childish personality. So, okay, so to sum up my talk. So we find out that when we have active fluid and we confine it into the water in oil droplet, and they can develop a spontaneous circulatory flow. And this circulation depends not only the droplet geometry, but also depends on oil configuration. And the reason we have this dependence is because the flow coupling between active fluid and oil across the water-oil interface. And this coupling was reproduced by a continuous active fluid simulation. And so with this knowledge, we developed a mini fluid device that allows us to turn on and off of the intra-droplet circulation in real time. So this is our recent exciting finding from our lab, and I hope you like and enjoy it, and thank you for your attention. And now I would like to turn it over to Ali Yusef. Ali, you can take over.
Hello, everyone. My name is Ali Yusef, and I'm part of computer science program. And my research focuses on developing new mathematical tools to better analyze neuroscience data. And today I'm going to show one of those examples where we are using mathematics and statistics in the development of new generation of treatments for psychiatric illnesses. I'm sure that you know the psychiatric issues is a common problem in our society, and there are many people who don't have a very proper treatment. Around $60 billion per year has been associated with disability in this domain. And at each moment of the time, where there is more than 1.5 million patients that they don't have a proper treatment. And the same number goes to anxiety and trauma disorder as well. Medication and talk therapy is effective in limited group of people. And the challenge that we are thinking might better address treatment is building a brain circuit specific therapy that can go to the actual cause of these illnesses. So let me explain it a little bit more here. So let's have two patients who are diagnosed with post-traumatic stress disorder. If we go over their spectrum of symptoms, they have completely different profiles. Some of them might have cognitive flexibility issues, some of them might have impulsivity issues, and some might have emotion regulation problems. Because of knowing that one, we have to realize that underlying circuit-based abnormalities vary from one patient to another patient. And so that suggests that the circuit that, so the other thing we know, the circuit which is underlying these individual symptoms are well characterized. As a result, assessment of each patient's symptoms of normality reveals personalized symptom domains that would be most advantageous if we can rectify that one. What's the outcome? If we know each of these circuits and through brain stimulation, we can bring abnormality to a healthy dynamics. We are building a personalized treatment for each individual patients. So let's put it in a very simple example. People might have problem with their cognitive flexibility. A person who might be rigid, no matter of the context of the day or situation, might take the left path, never take the right path. But under different circumstances, maybe we need to take the right path. Let's assume there is an accident on the left side, there is a threat on the left side. A person who is very rigid doesn't care about what's happening there. It's always picking that one. We are hoping through a treatment, we are giving other options to people who might face the rigidity problem. And the same statement can go to impulsivity, it can go to emotion regulation as well. So what we have taught here, what we are taking as a next step, if we know this specific functional domains, maybe we can pick a proper cognitive experiment. As we are running this experiment, we can collect both behavioral data and neural activity. And through the mathematics and the statistics, what I brought on the table was a way to characterize the relationship between behavior and neural activity in search for cognitive flexibility. If we can build this association by having a model which can trace dynamics of healthy and pathological brain states, we can know at which moment of the time the person is derailing from a healthy cognitive flexibility or other functional domain. And we can bring the stimulation and take that neural dynamics in a healthy range. So let's see what happens. Actually, as a part of my research, I was able to bring this one from a theory to the practice. What I'm going to show here, so probably I'm going to show this graph to you, which explains what we have done through our experiment. The theory of mathematics now comes to practice. And as you might see, we have a patient on top runs the task that I described for you. In the meantime, we are recording brain activity, and we are able to trace what's the cognitive flexibility of the patient over time might be, and inject proper stimulation at specific time points, which brings the brain dynamics to a healthy range as well. So what's the outcome of that one? If we are building these things, we can help patients who are suffering from different domains of psychiatric illnesses. This is one example that I'm thinking.
Any change in your depression since you had the deep brain stimulator?
Significant. It was my last hope, and it's, you know, with the exception of a few ups and downs, it's been a lifesaver. I feel much more just normal around them, and I'm actually starting to look for work now, or contemplating it. And my ideal would be to sort of work towards the end of the summer. It's a little bit scary, but at least I'm thinking about it, so it's exciting.
The big picture of my research, I'm doing interdisciplinary research, and WPI has a very vibrant environment for that one. And we are collaborating with neuroscientists. And as an engineer and mathematician, I'm making a bridge of using engineering in those domains that I discussed to be possible. Under the leadership of Dean King, we are taking another domain of research as well, which is focusing on pain. And I'm thinking we can bring the same knowledge that I explained previously to this domain. I'm hoping by developing new algorithms and tools, we can help human health and their life prospect. I would be happy to share the research that I've presented here will be published in Nature Biomedical Engineering very soon. And I'm gonna stop here. I pass to my colleague, Elizabeth Ryder, who is gonna talk about her research as well.
Thanks, Ellie. So today I'd like to talk with you about two intertwined projects, the BioCS Bridge Project and the Beacology Project, that allow us to bridge biology and computer science to engage high school students in solving real-world problems. So I work on a team, Carolina Ruiz in computer science and Sherry Weaver at the STEM Education Center at WPI, and with Rob Jagir, who's an ecologist at UMass Dartmouth. And you can see our teachers, our 13 high school teachers that teach at 8 different schools across central Massachusetts. So the problem that we're interested in is how to integrate computational thinking into high school STEM curriculum. So why would we be interested in that problem? So we all know that computer science and computational thinking play a critical role in every aspect of our digital society and are integral to modern science. So it follows that computer science should be accessible for all K-12 students, not just those taking elective computer science courses in high school. Computer science practices should be an integral part of the K-12 STEM curriculum. So things like data interpretation, abstraction, modeling, and simulation, and so on. So we really need a true integration of CS practices into K-12 STEM courses. So our team decided to approach this by forming a transdisciplinary team made of university faculty and students who contributed the research motivation behind our curriculum, high school faculty and students who contributed pedagogical expertise and kind of a test ground for our curriculum. And individuals in both of these groups, of course, contributed biological as well as computer science content knowledge to create the curriculum. So the motivational research problem underlying the curriculum is pollinator decline and loss of biodiversity. So we know that pollinators and plants are keystone species in the ecology, and that means that the ecosystem will essentially fall apart without them. So we know that pollinators are essential to the reproduction of flowering plants, and these plants in turn provide food and shelter to other organisms at higher trophic levels in the ecosystem. We also know that biodiversity is critical in the maintenance of resilient ecosystems. And so with these ideas in mind, Rob Jagir spearheaded the Beecology Project, which is a citizen science project with the goal of gathering information on our native bumblebee pollinators and the plants they forage on and pollinate to inform policymakers so they can better conserve and manage our ecological resources. So the team came up with a suite of software tools that are used by both the Beecology Project and the BioCS Bridge Project, starting with our web app, which allows the citizen scientists to identify different bee species and the flowers they're foraging on. They can then submit them to these observations to our Beecology database. We have a whole suite of interactive data analysis and visualization tools, as well as modeling and simulation components that can be used for hypothesis testing. And all of these tools are publicly available on our Beecology website. So students in our modular curriculum can learn to use these tools as well as building their own tools to understand and address real biological problems. So our curriculum contains 3 biology units, 4 computer science units, which are tightly connected. And the curriculum is aligned with Massachusetts K-12 CS and ecology standards. So teachers and students can use the curriculum to learn to integrate computer science and STEM practices to solve real world problems. So they can use our Bee Flower ID web app to contribute to our database as citizen scientists. And then they can visualize and analyze real data that they've actually contributed to building this database to test hypotheses. So they can look at things like diversity across the state. So a deeper color means more diverse, more different bee species. They can look at patterns of which species of bees are attracted to which species of flowers. They can look at the time course of bee species over time to look at population decline in different species. In addition, they can learn to write their own Python code to delve more deeply into the database and look for patterns in the data. So in this example, they can look at the tongue length of different bee species and see how it corresponds to the flower shape due to the coevolution of bees and flowers. And finally, students can learn to use and build simulations to test hypotheses. So in this simulation, the bees are represented by these pyramids, the flowers are the spheres. You'll see the bees pollinating the flowers and they carry pollen, changing color. The mice eat the seeds generated by the pollinated flowers, and you saw the hawks eating the mice. And so students can interact with this simulation and look at the different populations as they change over time. And they can also use it to test hypotheses. So if the ecosystem, for example, has too few of the bumblebee keystone species, it's going to be unable to support the top predator, and the hawks will vanish from the ecosystem. So I hope I've convinced you that our curriculum engages students to better understand both computer science and biology, and to learn to use and build computer science tools to solve important biological problems. I think in the interest of time, I'll skip over our teacher testimonials and go right to my thank you slide to the many students and teachers that have contributed. The ones in red are the students that we have working with us this year. And we also gratefully acknowledge the support of the NSF who funded our projects. And thank you so much for listening. And now I'll turn it back over to Craig.
Okay, thank you very much all. well, on one hand, I appreciate that everybody kept to the spirit of our lightning talks here. I always say a good faculty member can talk about any topic for any amount of time, but talking about a topic for a relatively short amount of time is actually harder. We did a pretty good job, but I don't know that we're going to have time for questions here. But I do again want to thank everybody. I know there are a couple of questions in the chat there, that one for Lindsey and one for Stacy. If you haven't looked, and I would encourage you to get back to the people directly. And if you're out there in the audience and have questions, certainly contact our speakers directly. I'm sure they'd be happy to chat with you. So in finishing up here, let me say a few things. So one, again, I encourage everybody to come back at 3 o'clock. We will have graduate student lightning talks. So come back for that. I want to personally thank Jean King here for starting Arts and Science Week here at WPI. It is a great thing, and it showcases the wealth and the range of things that happens here in our school. And I absolutely want to thank Rebecca. Without Rebecca, well, these wouldn't be happening. While I get to have the speaking parts and move you along here, Rebecca is the one that makes things happen here. So thank you, Rebecca and Jean's staff. Okay, so with that, I hope to see you all again at 3 o'clock. And thank you all for your presentations. All right. Bye.